7 decades since the Dead Sea Scrolls were discovered, around 80 new fragments of the ancient texts have been revealed to the public.
Portions of the Dead Sea Scrolls are here at DC's Museum of the Bible, removing all obscurity and open for all to see.
For 70 years, the Dead Sea Scrolls sat at the center of one of the greatest debates in the history of religion and archaeology. Brilliant scholars deciphered ancient scripts, reconstructed damaged manuscripts, and built elaborate theories about who wrote these texts and why they were hidden. Then somebody asked a question nobody had thought to ask. Not what do these scrolls say? What are they made of? In a laboratory at Tel Aviv University, a geneticist named Oded Rakavi extracted ancient DNA from the dust clinging to a single scroll fragment, ran the genetic sequence, and stopped. Because the animal whose skin had become that piece of parchment could not have lived anywhere near the cave where it was found. Not for a season, not for a week, not for a single day. And what that one impossible result quietly proved is going to change everything you thought you knew about the most significant archaeological discovery of the 20th century. The shepherd and the cave. To understand why that result mattered so much, you have to go back to the moment these scrolls first entered the modern world. In 1947, a young Bedouin shepherd named Mohammed Edge Dib threw a rock into a cave near the Dead Sea and heard something shatter inside. He climbed in. He found ancient clay jars stuffed with leather scrolls, each one carefully wrapped in linen. He had been looking for a lost goat. He had no idea what he was holding. Nobody did. Those scrolls turned out to be the most significant archaeological discovery of the 20th century. The oldest known biblical manuscripts in existence, dating back more than 2,000 years. Between 1947 and 1956, roughly 25,000 fragments, representing approximately 1,000 manuscripts, were recovered from 11 caves near the ruins of Qumran. They contained the oldest known copies of biblical books, Genesis, Isaiah, Psalms, alongside sectarian writings, community rules, hymns, and apocalyptic prophecies spanning roughly 250 BCE to 68 CE, the most turbulent period in the history of both Judaism and early Christianity. Sealed in clay jars in the dry Judean Desert, they had outlasted everything around them. The Temple in Jerusalem, the Roman Empire, every civilization that rose and fell across 20 centuries. But when scholars finally unrolled them, the scrolls didn't just offer ancient texts. They raised questions that would take 7 decades to begin answering. The theory that ruled for decades. Scholars from universities and seminaries around the world studied every visible feature of the scrolls. They examined handwriting styles, parchment color, texture, and thickness. They analyzed language, theology, calendar systems, and literary structure. And the dominant theory they arrived at pointed to a Jewish sect known as the Essenes. Ancient historians, including Josephus and Pliny the Elder, described the Essenes as a monastic community living in isolation in the Judean Desert, practicing strict purity laws, copying sacred texts, waiting for divine judgment to descend on a corrupt world. The settlement at Qumran, just a short distance from the caves, looked exactly like the kind of community those historians described. The sectarian writings within the scrolls described a tight-knit religious group with its own hierarchy, its own rules, and its own theological calendar based on the sun rather than the moon, a direct challenge to the Jerusalem priesthood. The famous War Scroll, describing a final cosmic battle between the sons of light and the sons of darkness, sounded precisely like the kind of text a separatist community operating at the fringes of society would produce. The conclusion felt inevitable. The Essenes had lived at Qumran, copied their sacred texts there, and when the Roman army swept through during the Jewish revolt in 68 CE, They hid their library in the nearby caves for safekeeping. For decades, this was the accepted story. Taught in seminaries, featured in museum exhibitions, repeated in documentary films, the Dead Sea Scrolls were the Essene library. Case closed. Stay with me, because this is where it falls apart. The cracks in the consensus. Even as the Essene theory dominated scholarship, uncomfortable questions kept surfacing that traditional methods could not resolve. The scrolls showed extraordinary diversity. Different versions of the same biblical books, texts written in different scripts by different hands. Parchment quality varied wildly. Finely prepared skins sitting alongside rough materials that looked almost amateur by comparison. Radiocarbon dating applied to select fragments in the early 1990s revealed that some scrolls predated the Qumran settlement itself. The numbers simply did not fit. And then there was the question nobody could answer satisfactorily. Why would a single isolated sect preserve multiple conflicting versions of their own sacred texts? The scrolls contained two versions of the book of Jeremiah, one matching the familiar Hebrew Bible, longer and arranged in standard order, and another significantly shorter with entire passages missing and chapters in a completely different sequence. For a community defined by strict religious discipline, maintaining contradictory versions of their foundational texts made no sense at all. Some scholars proposed alternatives. Perhaps Qumran wasn't a monastery. Perhaps it was a pottery factory, a winter villa, a military outpost. Perhaps the caves had functioned as a sacred storage space where Jews from many different communities deposited worn-out texts that religious law prevented them from simply discarding. These theories existed, but they lacked the hard evidence needed to dislodge a consensus that had been building for half a century. Then something completely unexpected entered the story. Not from an archaeologist, not from a theologian, from a geneticist eating lunch. The conversation that changed everything. In 2012, at Tel Aviv University, a molecular biologist named Oded Rakavi sat down to lunch with a biblical scholar named Noam Mizrahi. Their conversation turned to the Dead Sea Scrolls, specifically to the problem of matching thousands of tiny fragments with no titles, no page numbers, and no reliable method for determining which pieces belonged to which manuscripts. Scholars were working from handwriting comparisons and educated guesses about parchment appearance. Subjective, imprecise, and prone to error. The stakes were enormous. The tools were entirely inadequate. Rakavi listened. Then he asked the question that changed everything. What are the scrolls made of? Parchment, Mizrahi told him. Animal skin. Rakavi paused. Then the parchment has DNA. That single sentence cracked open an entirely new field of research. Rakavi specialized in ancient DNA analysis, a technique that had already transformed archaeology by revealing human migration patterns and population history across thousands of years. He immediately understood that nobody had ever attempted to apply it to manuscripts. Together with Penina Shore, the curator of the Dead Sea Scrolls at the Israel Antiquities Authority, Rakavi and Mizrahi began assembling the team that would spend the next several years attempting something with no precedent. Reading the DNA without touching the scrolls. The first obstacle was seemingly insurmountable. The Dead Sea Scrolls are sacred texts revered by multiple religious traditions and irreplaceable by any measure. Any technique requiring cuts, drilling, or sample removal was forbidden. The scrolls had to remain completely intact. How do you extract genetic material from something you are not allowed to touch? The answer had been sitting in the storage boxes the entire time. Over decades of careful scholarly study, tiny particles of parchment had accumulated as dust in the containers where scroll fragments were kept. That dust contained collagen, the structural protein of animal skin. And embedded within that collagen, preserved across 2,000 years, was ancient DNA. By collecting this dust from blank margins and damaged edges where no text existed, the team could gather enough material for analysis without ever touching a written surface. The answer was in the dust all along. Extracting usable genetic sequences from that dust was another matter entirely. The ancient parchment process itself, soaking, scraping, stretching, drying, damages DNA at the molecular level. Iron-based inks are chemically corrosive. The scrolls then spent 2,000 years in desert caves, followed by 70 years of handling by researchers who each potentially left traces of modern DNA on the surface. Separating ancient animal sequences from everything else required working in specialized laboratories under conditions resembling a sterile clean room, full protective suits, positive air pressure, ultraviolet sterilization, and developing strict authentication protocols to confirm that every recovered sequence came from an ancient animal rather than a modern hand. It took years, hundreds of samples, multiple failures. Eventually, reliable genetic sequences from dozens of Dead Sea Scroll fragments were in hand, and when Rakavi examined what those sequences revealed, he reached for his phone. The cow that broke the theory. Most fragments came back exactly as expected. Sheepskin. Sheep were common throughout ancient Judea. Sheepskin was the standard parchment material. Sheep could survive the harsh desert environment around Qumran, but some fragments came back as cowhide. And that result was not just unusual, it was physically impossible. Qumran sits in the Judean Desert, near the Dead Sea, one of the most arid, inhospitable environments on the planet. Sheep and goats survive there because they are adapted to extreme conditions, minimal water, and sparse vegetation. Cows cannot. Cows require dozens of gallons of water per animal every single day. They need substantial grazing land with real grass. There was no way any community living at Qumran could have raised cattle. Cow parchment cannot be produced in the Judean Desert. Any scroll written on cowhide had to have come from somewhere else entirely. When Penina Shore saw that result at the Israel Antiquities Authority, she stopped. She had spent her career as custodian of these fragments. She knew every box, every edge, every damaged corner. She read the result twice. Then she told her colleagues that this was the moment she understood everything was about to change. Not just the academic debate about the Essenes. The entire framework for understanding what the Dead Sea Scrolls actually are. And the cowhide was not random. It was attached to specific texts, specific versions of specific books, which is where the DNA evidence becomes something extraordinary. Fingerprints in the parchment. By analyzing genetic relationships between different parchment samples, researchers could determine whether multiple fragments came from the same individual animal. Two pieces of parchment made from the same sheep share essentially identical mitochondrial DNA. Two pieces from different sheep do not. For 70 years, matching fragments had been guesswork. Now they had genetics. They had fingerprints. Take the book of Jeremiah. The Dead Sea Scrolls preserve two dramatically different versions. One matches the Hebrew Bible familiar today, longer, chapters in standard order. The other is significantly shorter, missing entire passages, chapters arranged in a completely different sequence. Scholars had argued for decades over what that meant. The DNA delivered a definitive answer. Fragments of the shorter Jeremiah were written on cowhide. Fragments of the longer Jeremiah were written on sheepskin. They came from different animals, almost certainly from different geographic regions. They were not varying copies of the same tradition. They were two distinct textual traditions developed separately in separate communities in different parts of ancient Judea. Two communities, two versions of Jeremiah, neither one agreeing on what the book should say. The team also examined kinship patterns, using nuclear DNA markers to determine whether parchments came from genetically related animals, siblings or members of the same flock. Clusters of related animals pointed toward specific production centers where multiple scrolls were made from locally sourced animals at roughly the same time. Some of those clusters were mixed with parchments from completely unrelated sources. Different animals, different flocks, no genetic connection whatsoever. The collection had not grown organically from a single scriptorium. It had been assembled from multiple origins, representing multiple communities across multiple decades and possibly centuries. The library theory did not just crack, it collapsed. A world of many voices. Here is what that means. If the scrolls came from one isolated sect at Qumran, their extraordinary diversity was almost inexplicable. But if the collection came from multiple communities across ancient Judea, brought together into the caves over decades, and finally in one catastrophic moment of crisis, everything makes sense. The diversity is not a problem, it is the whole point. The DNA evidence provided physical support for a theory some scholars had proposed but could never prove. The caves near Qumran had functioned as a geniza, a sacred storage place where Jews from various communities deposited worn-out or theologically sensitive texts that Jewish law prohibited from simply discarding. Any document containing the divine name must be stored with reverence, not destroyed. Ancient communities maintained designated spaces called genizot for exactly this purpose. Jerusalem stands only about 20 miles from Qumran. When the Roman army besieged the city between 68 and 70 CE, Jews fleeing the catastrophe may have carried sacred texts into the desert, adding them to materials that communities had been depositing in the caves for generations. Family scrolls, synagogue scrolls, the accumulated written memory of a world that was ending. The caves near Qumran were not a monastery's private archive. They were a sacred library belonging to all of Israel. The Cowhide fragments proved something more. Texts were physically moving between regions. Someone had carried scrolls written on cow parchment from wetter, more fertile territory north or west of the desert into the Judean wilderness. This was not a closed community looking inward. It was a connected network of Jewish communities exchanging texts, ideas, and religious life across the ancient landscape. And here is what that means for understanding the world that produced early Christianity. The environment in which Jesus lived and taught, in which the first Christian movement took shape, was not one of theological uniformity. It was a world of vigorous, geographically diverse religious debate. The Dead Sea Scrolls did not belong to one faction in that debate. They captured the whole argument. A Bible that was still being written, a tradition that was still contested. The process of canonization was ongoing, still unresolved, different in different communities, in different places. The evidence had been sitting in the dust on a shelf the entire time. The story, written in the skin. The 2020 publication of this DNA research opened an entirely new era in Dead Sea Scrolls scholarship. Perhaps more remarkable than what it answered was what it made possible to ask for the first time. Can researchers identify specific production centers for different texts? Can they map the geographic network of ancient Jewish scribal communities? Can they track how textual traditions moved through Judea over centuries? Researchers are already pursuing these questions, analyzing DNA from a much larger sample of fragments, combining genetic data with multispectral imaging that recovers text invisible to the naked eye, building databases of ancient animal DNA from across the Near East, working toward a geographic reference map precise enough to identify roughly where in the ancient world an animal lived whose skin became a sacred text. The work of reading the Dead Sea Scrolls is not finished. It has just begun. For 70 years, brilliant scholars studied these texts with every tool available to them. Their work was real and irreplaceable. But there were questions that handwriting analysis could not answer. that parchment comparison could not resolve. Molecular biology provided a different lens. By reading the genetic information preserved in the very material the words were written on, scientists answered questions that had seemed permanently beyond reach. What that evidence revealed was larger than what anyone had hoped. The Dead Sea Scrolls are not the theological statement of one radical sect hiding from Rome in the desert. They are the accumulated sacred literature of ancient Israel. Multiple voices, multiple communities, multiple versions of the Bible, each one carried by someone into the caves of the Judean Desert, when the world they knew was ending. We discovered this because scientists learned to read the story written not in ink on the surface, but in the DNA of the animals whose skin carried that ink across 2,000 years of silence. In 1947, a shepherd's rock broke a clay jar and let the ancient world speak again. In 2020, Oded Rakavi sequenced DNA from ancient dust. The shepherd opened the door. The geneticist heard what the door had been waiting to say. If this kind of story moves you, the kind where one question asked over lunch leads to the rewriting of history, subscribe right now and turn on notifications, because we are just getting started.